lately with Pennsylvania, and he's got, what, 50 years experience, 40? It's like, yeah, but he's got some different ideas on CWD and what his thoughts and experiences he's going to relate to us with his program, and I guess I'll just let you kind of give your own introduction here.
Well, I'm a MD, neuropathologist, and trained at Duke, and I've been searching for the cause of the TSEs for my career, and I think we've got it. But anyway, the TSEs, you know, you're all centered on chronic wasting disease for an obvious reason, because there's an epidemic going on. And I would assume that if it wasn't for the epidemic, my stuff probably would have died on the vine because of the prion stuff. But anyway, much of my career I've dealt with Kreisberg-Jakob disease. This is a devastating disease. It affects an individual in the prime of life, and then they develop a rapidly progressive dementia, ataxia, and maybe in a few weeks they're dead. Okay. Now, the pathology, I don't need prion to make the diagnosis. I can look at the slide and see the characteristics, the spongiform changes in the brain. But of course they're prion positive, and the prion, obviously is used as a diagnostic tool post-mortem, but it's a devastating disease. And if you're, right now there's no diagnostic test. Somebody comes in with dementia, and it's usually after the fact, maybe months after the fact, that the final diagnosis is made. So we need a test, we need a diagnostic. And so you want to catch them early. I mean, possibly you could do something if you caught it early enough. And the patient, the disease is, as I say, devastating, and they say it's rare, but my gosh, I found 7 cases in New Orleans one year, so it's not that uncommon. And I think a lot of the problem is many general pathologists really don't know how to make the diagnosis. So as a neuropathologist, I have a better handle on that. Now, it was mentioned how long scrapie has been known, over 300 years, I think. Mad cow disease in Europe, actually in Europe, but basically happened in Canada. We've had a few cases in Alabama. You know, I spent 20 years as a neuropathologist at the University of South Alabama. Transmissible encephalopathy is considered to come from sheep scrapie. Then there's chronic wasting disease. And TSEs, or transmissible spongiformencephalopathies, in exotic animals from zoos, and cats are susceptible. Actually, the original diagnosis of mad cow disease in Europe was in cats from being fed contaminated food. Now, here's the latest one I could find. It's in Quebec now. I'm originally from Saskatchewan, and I think some of the researchers up there indicated that we may have spread it to Korea. But anyway, you can probably blame the English for spreading scrapie all over the place. But anyway, this is the devastating distribution of chronic wasting disease. Now, there is a transmissible agent in chronic wasting disease, any of the TSEs, and it's filterable. That is, it will not go through a 100 nanometer filter. It probably is in the size range of about 40 nanometers. Now, what size is that? Okay. A herpes virus is 120 nanometers. Okay. I've measured the bacteria, and as low as 40 nanometers, but I'll show you that later. So it's tiny. It's viral-like in size, and it's associated with prion protein deposition. Long incubation. You know, they argue, well, what about this long incubation against this bacterial idea? What about syphilis? You know, there's lots of bacterial diseases out there that have long incubation. Minimal immune pathology. I'll show you sections of tissues that don't show any immune reaction to the organism. Now, this caused all the problem. In the 60s, the transmissible agent, and these were studies with scrapie transmission to mice, was that the organism, the agent, was very resistant. You could boil it, you could show resistance to radiation, you could put it in formalin, couldn't fix it. And of course, that was even, they were using tissue homogenants. And the point is that this extreme resistance caused the whole prion concept. Basically what happened is that a mathematician looked at this data, this resistance, and he came up with the idea that, well, it couldn't be a virus, it couldn't be a bacteria because of this resistance. For example, resistance to autoclaving. So he said it had to be a protein. Well, wait a second, what about Pasteur? Pasteur came up with the germ theory, or the germ evidence of the germ theory, based on the fact that based on the fact that the proteins don't replicate. They don't replicate. And he supposedly spent much of his career proving that. Now, susceptibility to tetracycline. Now, basically, you can take experimental scrapie in mice, and you can treat with, at the same time you inoculate the animals, you treat with tetracycline, and you can actually prevent the onset of the infection. So whatever is causing it is susceptible to tetracycline. The problem with tetracycline is a bacteriostatic drug. It doesn't kill anything, and so that is a problem. Now, back in 1976, I looked at a brain biopsy from a patient with Creutzfeldt-Jakob disease, and I found by electron microscopy the presence of a coiled bacteria. And it come to find out that this organism caused, the rabbit tick isolate caused the Spongiformencephalopathy in mice. So I looked at, here's the brain biopsy and a typical Spongiformencephalopathy, but you can see the coiled structure within the neuronal process, and you can see the similarity to the spiroplasma. Here, this is a rabbit tick isolate in broth culture. And so when we did a study in collaboration with the NIH and inoculated the spiroplasma, this lab strain, into rats, suckling rats, and produced a Spongiformencephalopathy. And if you look at cultures of these organs, here's a pellet of a culture of a, spiroplasma. Note that, you know, there are filamentous forms, there's some coil forms, but a lot of these are bleb-like forms. So it's a multiform organism, and that's what's made it so difficult to detect by electron microscopy in the tissues. And the coil form is quite rare. Here in an infected rat, you can see a coiled spiroplasm, but that's quite unusual. Now, here is spiroplasma in broth culture, and here we are. Note this is an interesting one. They tend to have a central dense body, and they bud from this body. This is, and if a culture is under stress, if you stress it by treating it with some chemical, all these buds disappear. Also, at every stage of the growth cycle, you see here's the usual spiroplasma, about 100 nanometers in diameter. But here are there's collections of these ones with dense cytoplasm that essentially only measure less than 40 nanometers in diameter. Now, prion is not present in all cases of TSE. In fact, in about 10 to 15% of cases, you do not see the prion protein. But scrapie-associated fibrils are present in every case. This is negative stain electron microscopy, and these fibril proteins, about 200 nanometers in length, twisted fibrillar proteins measuring only about 4 nanometers in diameter, so they're very tiny. And these are absolutely diagnostic of any of the TSEs, including CWD, CJD, etc. Now, what's fascinating, if you take a spiroplasm, these are prepared by digestion of the scrapie tissues with proteases. Now, if you take a spiroplasma and treat with the same you end up with these exact fibril proteins within the organism. These are part of the structure of the organism that causes it to move. They move in a kind of a jack-in-the-box fashion. Also, scrapie antibody specific for scrapie reacts on Western blot with these proteins from spiroplasma. And here to the left is the spiroplasma proteins reacting on Western blot. And also the right is the CJD material. Now, Terry Spraker gave me some specimens of lymph node and brain tissue from CWD infected deer. And when we examined this with a polymerase chain reaction, it's a method to detect small amounts of nucleic acid. And it's very specific, it's so-called southern blot, where you also hybridize a primer to that PCR product. We were able to detect here in 7 cases of what was referred to as CWD, the presence of this spiroplasma bacteria. Now, this is just shows that there was adequate DNA present. The control samples were entirely negative. Now, what has allowed me to proceed in doing this work is I'm able to isolate the spiroplasma from CWD, from scrapie, and from Price of junk disease. I can actually grow it in culture and actually put that on agar plates, and it forms little colonies. Now, initially, we've been over many years been able to grow the organism out of scraping material, but it wouldn't propagate. But here's a material grown out of eye tissues from sheep with terminal scraping, and you can see the spiral in that that media by dark field microscopy. But it wouldn't propagate. Now, more recently, using a Bacilla media, which actually has sodium bisulfite in the medium, which lowers the oxygen tension, the organisms are culturable. This should be, this movie's not working. I don't know if we can or not. You can actually see the move in the, I don't know if, there we are. Perfect. So you can see the organisms that we've isolated out of the tissues, and they'll move all the way across the field. Now, take a look at this one. This is a large one with that central core and budding parts off the organism, which represents one of the complex forms of the spiroplasm that I showed earlier. Now, when you put it on agar plates, the spiroplasma produce specific colonies. They're subsurface colonies. That is, here's the surface of the agar. They grow into the agar, and they don't produce much on the surface of the agar. And this is classic for spiroplasma colonies. You can't make it out real well, but I have a section to the right here. That doesn't show up real well, but basically, if you stain this with oil red O, all those colonies are pink, and the oil red O will stain the organisms in the colonies. Now, what we did here was to scrape the surface of the colonies and then put it on a glass disc, and then we examined this by scanning electron microscopy. And basically, you can see the spiral nature of the organism. Phenotypically, this is a spiroplasm. And here's a clump of twisted organisms that came out of one of those colonies. So clearly, the organism is a spiroplasm. Now, we inoculated deer with the laboratory strain of spiroplasm.
Yep.
Note the bruxism or the movement of the mouth. This is classic for brainstem lesions.
No, I didn't get that. Would you care to move her forward a little bit?
Okay. Did you get it, Flo?
Hmm?
You got bad diarrhea right now.
And basically, this is classic for chronic wasting disease. Actually, I was at a meeting in Montana, and there was a group from Colorado presented the movie of chronic wasting disease in deer. A group from Idaho showed movie of Scrapie and sheep, and I showed the movie of the spiroplasma infection in the deer, and they were all essentially identical. Now, what we see here, here's the animal that's been affected. That occurred about 5 months after inoculation. There's 4 other animals in the group. They looked entirely normal, but they eventually all came down with it. So it's, you know, these animals obviously had organisms in their bodies, probably in their brains, but they still looked normal and eventually died of the disease. Now, when we looked at the tissues for these animals, there was a spongiform change here in the cerebellum, here in the cortex. But in the brainstem, or in the orbix, you saw the classic lesions, vacuolization of the neurons. And of course we had antibodies to this organism since it was the laboratory strain. And basically this brown staining represents the organism by immunohistochemistry. And here we demonstrated also in that same area the organism by electron microscopy. Now, what we found out in these animals that were infected with the laboratory strain was that there was a retinopathy consistently found in these animals. And that's true for Scrapie and sheep. It's true for CWD in humans. There's a retinopathy. And what was fascinating, and by the way, the arrows point to the spiroplasma in the tissues because we have antibodies to that particular organism. But look at the vitreous. The vitreous is the material that fills the whole posterior part of the eye, and in all of us. And here we have it looked like a pure culture of spiroplasma with absolutely no inflammation. And the arrows point to the organism. Now there's been, you know, you've heard about iatrogenic spread of Price-Reject disease by corneal transplant. Well, here we're looking at the posterior aspect of the eye. Here's the Descemet's membrane. Here's the corneal this is the corneostroma. But look at the endothelial cells lining the posterior aspect of the Descemet's membrane, and they're heavily infected with the organism. So I think this probably explains how transmission occurs from iatrogenic spread by corneal transplant. Now, this is a particularly exciting slides for me because basically, while I was at Tulane, I was able to isolate the spiroplasma from tissues, CWD tissues, and scrapie tissues and Kreutzfeldt-Jakob disease tissues. And what we used was passage in embryonated eggs. And so we prepared the material from the tissues, spun it down, and then put that supernatant in the eggs and incubated for about 8 days at 37 degrees centigrade. And when we opened the egg up, there was no embryo, just a slurry of material. And we took that and prepared that in culture, and then we inoculated that material into sheep and goats intracerebrally. And the sheep and goats developed classic lesions in the obex with these vacuolated neurons. And here's an example of scrapie and classic scrapie in the obex. These vacuolated neurons are essentially exactly the same. So this really shows that the inoculation of this spiroplasma isolate from these tissues, both with scrapie and also with chronic wasting disease, causes the lesions associated with scrapie and CWD. Now, here's what we had: these tissues frozen away. This study was done in 2007, and we had these tissues and frozen away. And then I developed a new methodology to use brucella media to make an isolation. So we went back to those tissues, brain and lymph node tissue, and was able to isolate the spiroplasma in cell-free culture. And here's a dark field study, and you can see the organisms, and you can even see the coiled organism. And you can see here's a negative stain electron microscopy showing the organism in the culture. So we have essentially completed Kochs postulates. Now, Kochs postulates determines causality. Basically, in postulate 1, you have to show an association of the organism with the disease. We've done that. I spent much of my career showing that association. So the organism, by PCR, by polymerase chain reaction, by electron microscopy, etc. Postulate 2, you have to grow it out of the tissues from that animal into cell-free culture, and we've done that. We've done that. Postulate 3, then you have to inoculate that material into a healthy animal and produce the disease. You've done that. And then you have to re-isolate the organism into cell-free culture from that experimentally inoculated animal. And we've done that. So we've essentially completed Kochs postulates of causality. Now, tissue culture. You can take the isolate from CWD and put into tissue culture. It'll grow. It'll produce some cytopathogenic effect, the vacuolization of here in a neuronal cell culture, here in a bovine corneal endothelial culture. And you can see by scanning electron microscopy the organisms on the surface of the cells. Here is a bovine corneal endothelial cell culture inoculated with spiroplasm.